Hi, everyone. Welcome to Geroscience Biotech and Health Extension Group, sponsored by 100 Plus Capital. Really happy to welcome you for the first event of this year. We're really excited to have Calen with us here today from Columbia University, and he is going to be discussing the effect of long-term caloric restriction on DNA methylation measures of biological aging in healthy adults from the CALERIE trial. I always love the abbreviations. Can't wait exactly what they stand for, word by word. But thanks a lot for joining us. I think it's a really good talk to have in January, as many people are trying to get on that train again a little bit. So you can tell us exactly the do's and don'ts and trying to help us make some sense of this very complex space. So thanks a lot for joining. Really appreciate it. Thanks everyone for being on, and I'm going to be in the chat monitoring for questions. So take it away. The stage is yours.
Great. So first of all, thanks so much for having me. These are really cool meetings and I've seen some great talks here. So I'm very thrilled to have an opportunity to join that list. Today, I'm going to talk about some work that I did when I first got to Columbia. This project is a big collaborative effort. The paper itself involves over 20 experts in areas as diverse as nutrition, molecular physiology, clinical trials, and caloric restriction. And the caveat right out of the gate here is that my background is not in caloric restriction. I was asked to take over the leadership of the project based on my background in DNA methylation and biological aging, and in particular in the area of epigenetic clocks. So I'm going to be talking about those today. And it was an exciting opportunity because it allowed me to shift my focus from areas that pertain more to how stress in the form of reproduction or inequality affect aging in the epigenome to a different kind of stress, and one that might even actually be beneficial, and that is caloric restriction. So I'm going to start with a fact that many on this call are going to be well aware of, and that is the idea that chronological age is basically the single best predictor of non-infectious disease, morbidity, and all-cause mortality. And as a simple demonstration of this fact, the figure on the left shows the log incidence rate for a range of chronic diseases. And you can see these sharp increases in many different types of diseases and morbidities across multiple organ systems, multiple physiological domains, and again, at the log scale. So these are really large increases with chronological age. To give you a sense of the relative contribution of aging, the figure on the right shows the contributing risk factors for the 3 leading causes of death in the United States in 2020. And you can see the disproportionate contribution of aging on the right-hand side of each of those 3 figures. And that contribution vastly outweighs other common risk factors like hypertension, smoking, and obesity. So we see these relationships, but we on this call certainly know that these diseases are not aging per se. So we believe them to be some manifestation of an underlying biological process. There is growing appreciation that damage is a primary driver of the biological process of aging. And that at the molecular level, this damage translates into a range of downstream molecular and cellular effects that are both conserved across phylogenetic taxa, from yeast to humans. And then we recognize those outcomes as hallmarks or pillars of aging. So this leads us up to the Geroscience hypothesis of aging, which is an overarching framework for understanding biological aging, where molecular changes shown at the left-hand side of the screen lead to declines in system integrity within and between organs. That eventually translates into functional declines. And then that functional decline eventually becomes disease, disability, and death. So with this foundation, we can then define aging as a biological process. It involves a gradual and progressive loss of system integrity. And it's a process that's also heterogeneous across species, populations, and individuals. Now, one of the implications of the Geroscience hypothesis is that aging starts far earlier in life than those inflection points for disease risk that I showed you in the first slide. And it suggests that the greatest potential for geroprotective interventions that would prevent those diseases and promote longevity might actually be earlier in life, before the onset of functional decline and disability. In model organisms, we're now at the point, I think we can all agree, where we have an arsenal of genetic and pharmacological tools for both geroprotection, and even rejuvenation. And that toolkit is only set to grow. And despite this toolkit and the amazing achievements that we've made in model organisms, in many cases, we actually still don't know whether or not, and if so, the extent to which those interventions translate into humans or to humans. Now, in some areas like genetic engineering, it's obvious why progress would be slow due to either practical or ethical concerns. But what's really surprising is that until recently, we've known very little about the actual impact of one of the simplest and most well-established seroprotective interventions, caloric restriction, and how that does or does not work to promote longevity in humans. Now, one of the challenges, as I implied, is a logistical one. And that's, how do we know if our intervention is actually going to slow biological aging and lifespan? And this figure shows sort of a hypothetical situation where you have decades of life on the x-axis, and then various states of health on the y-axis, with the blue area representing health span or healthy living state, and red morbidity. Obviously, it's a simplified line there. And then two different types of agers represented by the blue solid line or the red dash line, where you can see that one is a faster ager and declines into morbidity more quickly. The other is a slower ager and takes longer to reach that point. But the point here is to show that even with an effective intervention, we may have to wait years or even decades to observe the treatment effects on morbidity and mortality. And that is, until very recently, been a serious challenge of trials of geroprotective interventions. And it really comes down to how we measure the biological aging process. There are two main categories that have been used to quantify biological aging. The first is hypothesis driven. And this method basically focuses on the characteristics that we associate with aging, like functional impairment, multimorbidity, frailty. And what we can do is we can pick the list of things that we're interested in, and we just sum them up either as an accumulation of deficits or as the number of systems that are on the verge of failure. And so this figure just shows, for example, frailty, which is a measure developed by researchers that shows basically the expected increase with chronological age. And these methods are actually quite effective in older populations. They're good at telling us someone's lifespan or longevity or survival, but they're actually show very low variability in young populations. So they're not very applicable to people who have not yet reached the point where they're showing evidence of those functional declines. Another problem with these methods is that the risk that we derive from those measures isn't actually apparent, usually until after the disease processes that have led to them are well established. Because they're measured quite late in life, that means that they likely have the lowest sensitivity to change, which isn't ideal for any type of geroprotective intervention. The other main category of tools for quantifying biological aging are data-driven approaches. And these can involve studying things to do with the physiology of organs and organ system function, things like MRI, VO2 max, insulin sensitivity, or they can be based on molecular measures, which would include the epigenome, transcriptome, proteome, or metabolome. Now, there are some benefits of these methods, and that's that they are generally highly variable across the life course, even in young people. They provide a preclinical assessment of risk, so people don't have to be sick or diseased in order for us to measure them. And because they occur earlier in life, based on the geroscience hypothesis that I showed you at the beginning, we think they may be more sensitive to preventative interventions. Now, one family of molecular measure that provides a proxy of biological aging that many here are familiar with are epigenetic clocks. Epigenetic clocks are surrogates of biological aging. They're based on DNA methylation. They're usually measured using microarrays, in part because microarrays are relatively cheap, they're scalable, and they provide consistent data more or less across labs. They're generally developed using some machine learning methods, such as penalized regression, elastic net, or similar. And they're designed to provide a surrogate of biological aging by predicting an age-related outcome of interest. That could be age, that could be survival, or it could be a system's decline. And now I'm going to talk about the three different categories of epigenetic clocks and how they may or may not be useful for studying an intervention like caloric restriction. Okay, so the first, sometimes called first generation clocks, are those that are trained to predict chronological age. And these include Horvath's pan tissue clock, Hannum's clock, the skin and blood clock, and many others. And in this instance, biological age is the chronological age at which your body would appear normal in the reference or training population. So as an example here, you take methylation in the training sample, train it to predict a chronological age, and then you apply that to your new sample. And does the person in your new sample look older than expected for their true chronological age or younger? So the predicted age above or below the chronological age would then reflect the biological age. And we all know these are groundbreaking clocks. They've really changed the game as far as quantifying aging. But they've in general shown sometimes weak or inconsistent predictions with morbidity and mortality in humans, which is often what we're most interested in studies of longevity and interventions. So to address this gap was the second generation of epigenetic clocks, which were trained to predict survival. And these were really built in response to the shortcomings of the first generation clocks. And what they do is they, again, use machine learning and try to predict time to survival, usually by way of some measure of blood chemistry. These include the phenoage and Grimmage clocks. And their measure is the biological age at which your biology predicted survival would appear normal in the reference population. So again, we have this figure for someone who's roughly 70 years of age. And this person on the top would have a higher chance of death based on their methylation than the person on the bottom. Not surprisingly, these show good, consistent predictions of morbidity and mortality. There are some limitations. The first limitation is cohort effects. Now, because cohorts are all born together, they actually often can share the same exposure history for things like pollutants, toxins, pathogens, or health behaviors. And if we are measuring chronological age or survival cross-sectionally from a study, an older group of individuals will have different exposures from a younger group of individuals based on which cohort they're in. So you can see an example of this shown here on the right. And in this figure, it's showing differences in the kinds of exposures for someone who was born in, for example, 1900 versus someone who was born in 1960. And these, we don't actually know how these might affect the methylome, but based on other research showing how what you could call the exposome affects the methylome, we think these could be important, and it's worth keeping in mind when we start thinking about using these clocks as ways to study biological aging. Another limitation comes from survival bias. So that comes from the fact that older adults are, by the very definition, successful agers. So when we train our models to choose CPG sites on some outcome associated with chronological age or survival, we might not actually be seeing signs of aging, but rather signs of the adaptive response to aging or resilience. So this is akin to Wald's observation that the areas of the plains in World War II that most needed the armored reinforcement weren't those ones with the bullet holes shown in red, but the areas where the bullet holes didn't exist because these were spots that were so fatal that they prevented the plane from returning. And you can think of, in our training data, you can think of these gaps without bullet holes as the poor agers who have been selectively removed from the population. And a final limitation to the first and second generation clocks is that they actually can't tell us when the age acceleration occurred. For example, if we have two individuals, one might look epigenetically older than the other, again, blue looking or red looking epigenetically older than the other in this case. But we don't know if those are established early in development, let's say gestation over here, or if they reflect changes that have been accumulated throughout the intervening decades prior to the intervention. And so in either case, these kinds of measurements of biological age could actually be less sensitive to interventions that take place over relatively short timeframes. A third type of epigenetic clock is found in Dunedin pace, which is trained to predict pace of aging as an outcome. In this case, pace of aging is a composite measure based on the rate of decline across 19 different physiological and organ system measures at 4 different time points between 20 and 40 over a 2 decades of time in a single same age cohort in New Zealand called the Dunedin study. Rather than acting as a clock, Dunedin pace attempts to quantify the rate of decline in system integrity. And so it acts more like a speedometer than clock. So here we can see system integrity. Everybody in this figure is declining, but some people are declining slowly and some people are declining much more quickly. So Dunedin pace estimates biological aging rather than just biological age and the rate of decline of system integrity relative to some reference population, in this case, the Dunedin study. And this measure has demonstrated both strong prediction of morbidity and mortality and also high technical reliability, which is really important. And I'll touch back on that briefly later in the talk. Importantly, the Dunedin pace also does not suffer from some of the limitations of the other clocks. In particular, there are no cohort effects since it was generated in a single year birth cohort. There's no survival bias since the measurements were taken when the cohort, as I said, was between 20 and 40 years old. So there's almost zero age-related mortality. And because Dunedin pace was designed to study change, there's less ambiguity about the timing of age acceleration. So returning to our hypothetical examples, in samples taken from a single time point, let's say this is an intervention here, age and grim age will regard the people represented by the blue and red lines as different in terms of age acceleration, whereas Dunedin pace will accurately or correctly identify them as aging at the same pace. All right. So we have now nearly 80 years of research showing us that caloric restriction, which is defined as macronutrient restriction with maintenance of micronutrient sufficiency, is one of the most well-established geroprotective interventions in model organisms. There's findings in yeast, flies, worms, mice, macaques, all of which support the hypothesis that CR has been linked to better health and longer lifespan. But until very recently, there was no long-term trials of CR in healthy, non-obese adults. And this was changed with the CALERIE study, which stands for, and you may not want to remember this, but comprehensive assessment of long-term effects of reducing intake of energy. See, I even can't remember it. Okay, so the design of the study was relatively simple. 220 individuals were randomly assigned to either CR, caloric restriction, or AL, ad libitum groups in a ratio of 2:1, 2 CR participants for each 1 AL participant. And these matchings were stratified across the study sites, sex, and body mass index stratum. So for each time a person was allocated to one group, 2 were allocated to the other group, the CR group that looked very much like them. The CR participants received an intensive behavioral intervention that was designed to result in a 25% CR reduction relative to baseline. And this intervention included a 27-day meal provision phase at the beginning, so they could get their head around what 25% reduction in caloric intake looked like. Individual and group sessions to help them work through the struggles of CR, portion estimating training, digital assistants that help them monitor their caloric intake, as well as training in both nutritional and behavioral topics related to diet and nutrition. Adherence to CR and the percent CR caloric intake achieved was assessed using doubly labeled water. So that allows us to measure total energy expenditure and also changes in body composition. And the duration of the trial for both CR and AL participants was 2 years. In addition to regular questionnaires and some blood samples for other biochemistry measurements, blood samples were taken at baseline before randomization, 12 months and 24 months, so 1 year and 2 years, that were ultimately used for DNA methylation to extract the DNA for DNA methylation. And the overarching hypothesis that we went into this study with was the idea that if CR is effective at slowing biological aging, then all participants will age biologically during the intervention. 2 years is a while, but the CR treatment participants will age more slowly. Okay. So we'll start more generally, and I'll just describe some of the findings of other studies before we get into the epigenetic clocks. We see that on the figure on the left, we can see that CR was achieved in the treatment group, shown up here on the right, but that adherence was not perfect. So 25% would be about here. Many people didn't achieve 25%. They did, on the other hand, achieve some CR, and that was about 12%, whereas the ad libitum group only achieved 2% caloric reduction on average. Nevertheless, despite the fact that they didn't quite reach the full reduction in caloric intake, CR treatment was associated with a significant and sustained weight change throughout the 2-year study. And that's shown by these open circles here on the bottom. During the study, there were a range of clinical and physiological impacts that were achieved. And these include things such as loss of fat, but not muscle or organ mass, improved mood, sleep, self-rated health, broad improvements in cardiometabolic health, metabolic adaptation, reduced oxidative stress, reduced inflammation, improved thymic function, and signs of muscle rejuvenation. Work by our lab also showed the effect of CR on measures of blood chemistry, biological aging. And these are measured using the Clamara-Duval method, a clinical version of the pheno-age method. So this is not the epigenetic clock pheno-age, and homeostatic dysregulation. And here you can see the ad libitum group over time. And here you can see the CR group on the bottom. And you can see quite clearly the pattern matching our hypothesized response, where the ad libitum group increases over the 2-year period, whereas the CR group shows slower or a reduced biological age. Okay, so we have a pretty good idea that there's evidence of better health and slower biological aging using clinical measures, even though people didn't achieve the full 25% caloric reduction. Now, for the question about whether or not epigenetic clocks are actually suitable for trials of geroprotector like caloric restriction. Now, I put up this modified version of a checklist that was generated by Justice and Krzyzewski, and we can walk through it. We know all of the clocks are feasible for clinical trials in older individuals, very easy to collect on a small amount of blood. We know that they're robustly associated with chronological age across cohorts. We also know that at least in the case of pheno-age, grim-age, and Dunedin pace, they're robustly associated with morbidity and mortality, but we don't know if they'll be responsive to CR. So to answer that question, we specifically focused our analysis on the 3 clocks, in particular, pheno-age, grim-age, and Dunedin pace. And specifically, we used the principal component-derived versions of pheno-age and grim-age, which are called the PC clocks. Those are developed by Higgins Chen and the Levine lab and are predictive of mortality, but they also show improved technical reliability over the original clocks. And I said I'd come back to this technical reliability issue. Now, technical reliability is essentially just the ability for us to accurately measure the same biological age in the same sample twice. So if you have the same sample taken at the same time, you split it up into 2 different, that's my automatic light. You split it into 2 different aliquots, and then you measure biological aging. You want to know that you're basically going to get the same measurement. You're not going to get wildly different measurement. And the principal component clock versions of this do a better job of that. And that technical reliability is particularly important in a longitudinal study, because if you think about it, error or technical noise in the assessment of biological age is compounded over 2 time points because we're trying to measure change. So if there's error here and there's error here, we have a lot more room to make bad estimates of the change that we're observing. And then we focus on Dunedin pace, which, as I've noted, is a predictor of morbidity and survival, also has high technical reliability. And that's in part because it was constructed specifically using probes that showed high repeatability in replicate samples. Okay, and here are the results. So on the top, what you're looking at is plots that show the mean at baseline, 12 months, and 24 months relative to baseline. So all of them start at 0, basically. And therefore, PC pheno age, PC grem age, and Dunedin pace. And these are shown, the CR group is shown in red, the AL group is shown in blue. And what you're looking at here are the effect size estimates based on the models. And those are standardized to Cohen's d. So they're standardized to standard deviations of the data. So if you see 1, that means a 1 standard deviation change in that biological aging measure over a year, for example. At the bottom, you're actually looking at the raw data for each of these same figures. And in this case, each individual is shown by a point and a line connected by points connected by lines. And then the box plots are just showing medians and 75th and 25th quartiles. So here's just a tabular version of the same models, which included sex, race or ethnicity, the study site, pre-treatment baseline chronological age, and the body mass index stratum. And to get a sense for what those effect sizes actually mean, because I guess you can clearly see that the age, PC grem age, and PC pheno age are increasing over the duration of the study. Dunedin pace for the ER group is slowed and that actually stays more or less the same as 0 statistically. The ad lib stays the same. But what that effect means is the decrease is about 0.25 standard deviations. And if we multiply that out, we can see that's equivalent to about a 2.25% slower pace of aging in the treatment group compared to the control group. That 2 or 3% slower pace of aging is small, but it corresponds to a reduction in mortality risk if you use other studies where mortality risk has been determined by as much as 10 to 15%, which, looking through the literature, we found to be equivalent in magnitude to the effect of a smoking cessation intervention. Now, one of the other things you might have noticed about this figure is there was a lot of individual variation here. And so we next wanted to test whether the percent of CR was achieved was related to the change in Dunedin pace. There's 2 ways we could do that. The first way, we simply stratified based on whether or not a participant in the CR group achieved less than 10% CR or more than 10% CR. And we found those that achieved lower than 10% CR had 0.2 standard deviation decrease in Dunedin pace. Those who achieved more than 10% showed a much larger 0.33 standard deviation increase in Dunedin pace. So that's one way to look at it. Another way is to quantify the true effect of CR through instrumental variables analysis method that estimates the effect of treatment on the treated, or sometimes called TOT. And this basically involves a multi-level model where the first level estimates the effect of treatment on actual caloric restriction achieved for each participant. And then the second level uses that estimate as a covariate to estimate the effect of CR achieved through the intervention on Dunedin pace. Using this method shown over here on the right, we can see that the effect of a relatively high dose, 20%, still below the aimed 25% dose, but basically the 75th percentile of CR that was achieved in the CR group results in roughly 0.4 standard deviations decrease in Dunedin pace. And if we estimate those effects out using the same method I described before, those are about equivalent to 3.6% slower pace of aging among the treatment group. Now, one of the challenges that many blood-based epigenetic measures face is cell type heterogeneity. And that's basically because the blood that we use for DNA to measure epigenetic clocks is comprised of a collection of different immune cells. Each of those immune cells are contributing DNA, and each of them has a distinct methylome. And we still don't really know the extent to which this immune signal might actually confound some of our epigenetic age estimations, although work on that is actually underway in several groups at least. So what we did was we included CD4 cells, CD8 cells, natural killer cells, and B cells and neutrophils, which were estimated bioinformatically using DNA methylation data itself. And we found that basically the findings were essentially unaffected by including cell composition. So this further supports the idea that we're capturing some molecular process tied to biological aging and not a change in the composition of the immune system. Okay, so to wrap up, epigenetic clock measures of biological aging do seem to offer new opportunities for the analysis of intervention trials. But if you start to look more closely, different measures seem to have different properties, different strengths, and different limitations. And it may be the case that pace of aging measures are more sensitive to the effects of short-term interventions. I think more studies are going to have to be carried out to see if that's the case. It's important to note that even in a highly controlled, intensive 24-month trial, the effect sizes were relatively modest, and future studies should plan for this kind of modest effect in order to be well powered enough to detect changes in epigenetic clocks in response to some intervention. But I think it's worth pointing out that even modest reductions in biological age, like of the magnitude that we described, could have profound impacts on population health. So they shouldn't be, I don't think, summarily dismissed. And finally, we have proof of concept that epigenetic clocks provide good surrogate measures for biological aging, but we still need long-term follow-up trials in order to definitively establish that. And in the case of CALERIE, a follow-up study led by Dr. Sai Krupa Das is testing if those short-term effects that we observed during the trial translated into longer-term reductions in age-related chronic diseases or risk factors. And certainly that's not the last word on the CALERIE molecular data. I think a growing number of researchers are poised to start tapping into this data resource. And we're currently preparing a molecular data set repository for the scientific community. That includes genetic variation for all participants, DNA methylation for both adipose and muscle, led by Steve Horvath, RNA for adipose, led by Kim Huffman, and RNA for muscle, led by Luigi Ferrucci. And as we continue to integrate these measures, I think we'll continue to gain insights into the molecular effects of CR in humans, and maybe even into the process of biological aging in humans more broadly. So with that, I just want to thank my collaborators and especially Dr. Belsky, Dan Belsky, who's the PI of this grant. And thank you for your time.
Wonderful. Thank you so much. That was a lot to digest. Really, really cool. Thanks for sharing all the info. We already have a few questions and comments in the chat. Do you feel like okay to get into them, or do you want to take a breather?
Okay.
Cool. Then with that being said, maybe that direct, whoever you are as a person, do you want to unmute first and ask? And then Carl, if your comments are relevant for the audience, then you can go next.
I haven't muted. That direct is one of my companies, and the way you set up these Zoom calls, I can't change my name to John Hemming, which is what my name is. And the basic question, is there not a big problem trying to measure the state of somatic cells in general from however wide a selection of white blood cells? So you take the blood and you take the white blood cells and you do the epigenetic analysis on the DNA. And I'm an acetylation person anyway. However, you can't really measure the kidney, the pancreas, the heart, just from the white blood cell.
Yeah, so I guess you're asking whether or not blood is a good tissue to try to understand aging in other organs and tissues. And I think it's a valid question, but I think it's also, I would argue it has a lot going for it.
If you don't mind, I'm a great fan of using blood biomarkers. I have an issue with using the methylated white blood cell, whatever they are, for the rest, because obviously in your blood, you get a lot of information. No question. I'm a weird individual who does weekly blood tests because I need to monitor what's going on. But yes, I do methylation tests as well. But I'm not sure you can pick up the state of the kidney. If we think, what is methylation?
I think probably my response would be, so what the work we're doing is generally not mechanistic in nature, right? So we're not actually trying to say that the changes that we're witnessing in blood are the same changes that are present in the kidney. What we're trying to say is, can you predict the outcomes in other organs? And it seems like you can. If you can predict dementia, maybe if you can predict dementia from DNA methylation in blood, that's a useful surrogate measure. We don't really know why. In a lot of cases, it's a black box. Now there are groups but I think there are groups and there are people who are trying to work on that problem. So Morgan Levine's group working on the systems age clock. There are people who are breaking down individual clocks and saying, how well can we actually estimate aging? And I think the question of heterogeneity in aging across organs is fascinating. Do you have some people whose heart is aging faster than their kidneys or vice versa? And do they lead to the same outcomes? I think that's fascinating. I think it's an area that merits more work, but I don't think it discounts the fact that a predictive measure is still of mortality or disease or cancer, whether or not we know why it's doing that. Some of us, like us in the public health schools, maybe are less interested in why it does it and just interested in how well it does.
You're on mute now. I will let you maybe make one last final comment to that, and then we move on to the second question because we have so many of them. But direct, you're still on mute. I'm going to ask you to unmute. We cannot hear you.
John, you're muted.
Okay. You're unmuted.
There you go.
No, I haven't unmuted. You wanted me. Oh, right. Sorry. I think the issue is if you're trying to pick up this is what is methylation. Sorry, my cat has just walked across the keyboard. Have you just silenced everything?
No, we can hear you. We can hear you.
Right. Sorry. Look, the cats want me to feed them, and they're not very pleased because I'm not feeding them. And I think the question is,
we just learned.
What is methylation? Is methylation merely an indication for any one particular cell what genes are not being expressed? And if it is such an indication, then how do we pick up from white blood cells what genes are not being expressed in? I've got the cats doing it again. They want to be fed. What genes are not being expressed in kidney cells or whatever? And this is really where the interesting question is, where the balance is between methylation and acetylation. And so that I would not deny there is some merit, predictive element from white blood cells on the general somatic health, because I think interleukin 10 is the key driver.
Yeah, I think ultimately the data and the population level data speak for themselves as far as the utility of the markers. The biology of the markers, I think, is a fascinating topic, and I think it's still up for debate, definitely, without a question. But certainly, I think it's not a topic that I'm prepared to debate.
I don't mind. I'm quite relaxed. I will move on and mute myself.
John, you haven't said whether or not you're familiar with the systems aging clock that Morgan Levine started at Yale and Raghav Sehgal continued that work as his PhD work, which is all about
the very system. There's one based on blood biomarker. So you've got Morgan. There's actually a lot of different aging clocks worked out on blood biomarkers, and they are useful things. So this is where you get into the debate about individual biomarkers, which is, I'm sorry about this.
No, we're talking about the systems aging clock. That's a methylation clock that measures the aging of 9 or so different organ systems, just from methylation done
from white blood cells. I think there is an issue, which is this basic question of what is methylation? Is methylation?
I guess my point is, I suggest that you ask your question in terms of a criticism of that clock, which is doing exactly what you're saying, is hard to do. But you have to specifically frame it in terms of that clock.
I think we can leave. I'm very happy to debate these, but I think let's leave it for now because it's a good debate to be had, a worthwhile debate, but not with the context of that specific.
Okay, good. Closing this down for now. And Carl, you had your next comment, so please make it now.
I just want to make a comment. This is great work. It's so great to see a clock and CR be combined. And I was so excited when that paper came out. I think some of the discussion after that paper came out was inappropriately narrow. And this is something I posted about recently on LinkedIn and Twitter about the one-mindedness of some people in the field about looking at rate of aging and not looking at reversal of aging or rejuvenation events as discrete things. And so in the context of these things, I think I would broaden the conclusion comment that you made, which is that rate of aging clocks are probably better at detecting changes in interventional changes in rate of aging that are, especially in the short term, because it would take a long time for an X% rate of aging change to accumulate to a point where a GrimAge style point of time estimate aging clock would have a significant enough difference. And that's why GrimAge didn't show a good result here, because there's just 12% CR over 2 years is simply just not enough time. But the point of age estimate clocks, like the Gen 2 clocks, like GrimAge, should be better than rate of aging clocks at detecting rejuvenation interventions. So ones where instead of you're adjusting rate of aging, but you're jumping backwards to an earlier state of biological age. So if we had good examples of those types of therapeutics, I think GrimAge would do better relative to doing the pace for them. There's a big difference between rate of aging modulator therapeutics and rejuvenation therapeutics.
Yeah, I guess that's an empirical question. And I imagine with the advances that we're seeing right now with rejuvenation, maybe not in humans, but I think maybe we'll start to see some of the answers to that question. The one thing that's worth pointing out is some of the recent work coming out of Adam Gladishev's lab on the damage and adaptage clocks. In theory, if you have changes in methylation that are arising both from the damaging properties of aging, as well as the adaptive responses to that damage, you could in theory not see a change because they're canceling each other out. You might be rejuvenating by getting rid of some of the damage associated changes, but not increasing the adaptive associated changes. And it seems plausible, at least, that you may not see those effects. So maybe clocks, the new damage, adaptage, and cause age clocks will be particularly helpful for starting to pull apart which sort of sub-compartments of aging we're actually capturing. Yeah.
Agree. It's a theoretical point, not a point that GrimAge specifically is going to be better. But if you do get a damage type clock, that's a point of age estimate, that's probably the right approach if you truly had a suite of rejuvenative therapies that really did jump you back 5 years or something like that. All right. Thanks a lot.
Oh, we have one from Varun, which is just that he would love to connect, work on something similar, at least related enough that he would love your email, and he didn't get it when you mentioned briefly earlier. So if you opened it, maybe you could share it to him either privately or in the chat. And the next question we have is from Julie, if you want to unmute.
My question is, if you could have any sort of data you'd like from a similar study to the CALERIE trial, what would you want to gather? What kind of clocks or measures would you want to study?
I don't know that I would necessarily, I think the next step in my mind isn't more or different clocks. It's different interventions, probably, I would say. I don't know how many people on this call have tried caloric restriction, but it's not easy. And there's a reason why most people couldn't achieve 25%. I think a really exciting area is time restricted feeding or intermittent fasting. So would that show similar effects? I think in CALERIE, it didn't seem to matter when people ate as far as weight loss, but I'd be really interested to see a good study of intermittent fasting over a long period of time. And I think that's probably just around the corner. And hopefully the work that we did and the effect sizes that we showed will form a bedrock for other studies to just point their designs.
So I have a comment about that because I think one of the things you said is subtly not quite right. CR is hard, but if you look at the rate of aging at baseline of the calorie study participants, it was much better than it was like 0.94 or 3 or something. That indicates that the calorie study participants at baseline were much healthier than the Dunedin pace cohort was. And so it might be hard to achieve 12% CR or more when you start with that baseline, but it's maybe not that hard to be as healthy as the calorie study cohort compared to the Dunedin pace cohort.
Case, I mean, I'm very cautious about translating these numbers between studies. There's some questions that we're looking at that we're interested in cross-population variability and in clocks and so forth. But a lot of my prior work has been conducted in the Philippines with people who live in very different socio-ecological and nutritional environments. And there's a lot we don't know about what a value of X means in the Philippines or in Tanzania or in Brazil or in the US. We know the reference population. And I think starting to expand the diversity of samples that we have, that we understand how these clocks work, what they predict and don't predict, is an important next step in the field.
All right, I'm going to ask my closing question now. I have a few more that we could get into, unless someone else who hasn't asked a question raises a hand, then we may have another minute to cover that. So just an open invitation. The one thing that people usually are pretty interested in afterwards, there's two parts to this question. First one is, if like someone new entering the field was listening to this, what's like a thing that you, like a challenge that you'd love to have solved, like something for perhaps someone new entering on a junior level to take on that would make progress in this field much easier?
I guess mine are selfish. I think I'd like to see these measures become cheaper, right? Right now, we work with clinicians in the medical center and so forth, and even $300 a sample is still tough to get numbers for. And I think it would, that's not my side of the world, but I would love to see them get better and better at running arrays, targeting specific things, high throughput, low cost. And I feel like there's probably a desire for that in industry and in academia. So I feel like we're heading in that direction, but it still surprises me sometimes the way that they design the arrays, and maybe just not everyone's thinking about aging as much as we are. So maybe that's why.
Yeah, who does want things cheaper and faster? I think that would certainly have really big multiplying factors. So thanks for that. And a few people in this community are working on that. So I think it's quite encouraging. And then the next one is, and that can also be very self-interested. In fact, it should be, if people in this room right now, or like people watching this afterwards on YouTube, could do something to help you guys in your next steps. Maybe you could elaborate a little bit on what like immediate future work would you like to be doing, and how can others like help support you along this path?
I don't have any specific thing for just our work, but I think especially in a community like this, where the focus is on geroscience, geroprotection interventions, I think I would probably promote the support for science in general, and also the study of aging in public health. So real people in the world and real interventions in the world, whether those are cash interventions or social policies or things like that. I think scaling these, it's cool to have 200 people calorically restrict, but I think the power comes if we can start to figure out how do we slow aging and help people's health and quality of life in the real world generally. And I think that will change a lot. So I think thinking in that big picture would be probably my ask.
Wonderful. I love it. Any final bits and pieces that you wish we would have covered, but we didn't? Cool. Thanks everyone for joining. That was really wonderful. Certainly learned a lot. And I think we can all be a little bit more informed about any of the New Year's resolution that we personally took on for our health as we move into January. So thanks everyone for joining. Thanks a lot, Calen. This was great. And I'll see you all for the next one.